പറഞ്ഞു എനിക്ക് മെസ്സേജ് കിട്ടി അവിടെ കൊടുക്കാന്ന് പറഞ്ഞു. Okay, shall we start now?
Yes.
Hello. Yeah, I'm sorry for the delay because election rallies are going on over here. Because, that's why a lot of commotion. I hope I'm audible.
You are.
Yeah. So today we have the 19th Pediatric Potpourri, and this time we are talking about thalassemia. Most of us are having a lot of patients with thalassemia who we are dealing with, and pediatricians and PGs need to know, because this is a case that could, hepatosplenomegaly is a case that all of us, all pediatricians, all PGs would get for their long case or OSCE. And therefore, we need to know a lot about thalassemia and blood disorders. And for this, we have a good panel of experts. Dr. Vikram Matthews from Mellow, who's done a lot of transplants for thalassemia. And so we have him as a lead discussant for this for today. We have with him Dr. Shaji and Dr. Shivram from Manipal Hospitals will be doing the transfusion bit, and Dr. Fauzia will be doing the follow-up of patients for repeated transfusions. So we'll have a session for about an hour, followed by question and answers, and then the quiz as usual. We will start with I'm sharing the slides. We will start with case presentation as usual. The case will be presented by, I hope you can see my slides.
Yes, we can.
We'll start today's 19th Potpourri with this case presentation, which will be, and Dr. Vikram Matthews will take us through a case that he's had at Vellore. We'll start with Dr. Vikram Matthews. Let me introduce you to him. Pediatricians, we're not very familiar with him because he's not a pediatric person. He's done his MD, DM, and FASC, the professor of clinical hematology and Department of Hematology in the Christian College, Vellore. He did his MD medicine from CMC Vellore in 1996. DM in hematology, clinical hematology from the same institute in 2001. Postdoctorate fellow at the Stem Cell Biology Section, Department of Internal Medicine, Washington, in USA in 2003. Bone marrow transplant fellow in the BMT unit, Washington University, St. Louis, USA, 2004. And is currently working as professor at the Department of Hematology, CMC. He's welcome DBT India Alliance Senior Fellow 2012 and 2018. And his areas of interest are myeloid malignancies and allogenic stem cell transplant. Over to you, Dr. Vikram, for the presentation.
Good evening, everybody, and thank you, Dr. Jason and the IAP body of Cochin for having us. I'm going to get through. We have a crowded program, and the objective, obviously, is to give the students and the participants a flavor of thalassemia and how we approach the diagnosis and management. So I'm just going to take a case, which is a 9-month-old boy born of non-consanguineous parents. They are actually Sindhis in origin. The child presents with very non-specific symptoms, but prior to coming to us, he was already transfused. The local hospital records say he had a hemoglobin of 5. We don't have too much details on him. And I present it specifically like this, but because this is often the scenario in which the child comes to you. Patient was sick, he was transfused, and he comes to you post-transfusion, and you have to make a call how you're going to evaluate him. He has an elder sibling who we later find out his hemoglobin is also 9. The family history, the details are there are relatives both in the maternal and in the paternal side who have been diagnosed to have thalassemia trait. Next slide, please. So besides anemia, the rest of the hemogram is unremarkable. When he comes to you, his hemoglobin is actually 10. His MCV is almost the normal range. And again, I stress this is not the normal parameters how a thalassemia major child will present. but this is the reality of how you will often see patients, and you need to how to approach them. This is a 9-month-old child, so the spleen is just palpable, liver is just palpable. Often when you see these children at an older age, they'll have a frank hemolytic facies with a prominent hepatosplenomegaly, short stature, and failure to thrive, etc. So in this particular scenario, it's very straightforward. There's no suspicion or there's no subtlety about it. It's a suspected hemoglobinopathy. How does one evaluate this case? What happens when patients come to you transfused? You can't interpret the indices. So do you wait for anemia to recur before start evaluation? To address some of these questions, we will have Dr. Shaji walking us through diagnosis. It's important to understand why the anemia in thalassemias happens only after 5 to 6 months of age. And when people do come with transfusion, how do we approach them? Dr. Shaji will walk us through some of the diagnostic aspects of thalassemias. Shaji.
So for molecular basis and molecular diagnosis of major thalassemias, we have Dr. Shaji RV, who the PhD, the professor of Department of Hematology, adjunct scientist, Center for Stem Cells Research, Christian Medical College, Vellore. He did his PhD at Christian Medical College, Vellore. Postdoctoral fellowship at the St. Jude Christian Children's Research Hospital, Memphis, University of Chicago. And his areas of interest are molecular basis of arthroid diseases and induced pluripotent stem cells for disease modeling. Over to you, Shaji.
Hello, Dr. Vikram Matthews talked about a case where the patient was transfused, and he was talking about how important is the diagnosis to be established before treating the patients. So I will come to the importance of molecular diagnosis of thalassemias. Before that, I'll take you through some introduction and background on thalassemias. As most of you know, thalassemias are quantitative disorders of globin chain synthesis, and these diseases cause imbalance in globin chain levels, and that's a major reason for this disease. There are two major types of thalassemias, beta thalassemia and alpha thalassemia. What happens in beta thalassemia is that the beta globin chain is defective because of the mutations that occur in the beta globin gene. And in alpha thalassemia, what happens is one or both the genes of alpha globin cluster, they will be defective, and that can give phenotype also. We'll be focusing more on beta thalassemia because that is the most severe type of disease that we see in our population. Rarely, there are two other types of thalassemias, and it's important to know they exist in a fairly good frequency, and we see patients with these diseases, and they can come with a phenotype similar to what we see in homozygous beta thalassemia, and they include delta beta thalassemia and hereditary persistence of fetal hemoglobin. I'll go through them in the last few slides. Just to briefly tell you the molecular basis of beta thalassemia, you can see here the first figure that you see on the top is the structure of beta globin gene, and it has a promoter and coding sequences which codes for the protein to be formed, exon 1 and exon 3, and there's a polyaddition. This is typical structure of a gene. And you can see mutations all over the gene, and they can be in the promoter, exon, anywhere in the gene. And there are more than 200 mutations that have been reported to cause beta thalassemia. And most of those mutations are point mutations. And deletions of large sequences, they are extremely rare. The left bottom figure that shows that the different aspects of gene expression, and if mutations occur in most of these important regions of the beta globin gene, it can affect the transcription, splicing, and translation, the usual molecular processes that occur in producing a protein. If these mutations occur any part of the gene, that can affect the beta globin expression. So on the right side, you can see that it's because the beta globin levels goes down, you know that the adult hemoglobin contains two alpha globin chains and two beta globin chains. When the beta globin chains are reduced or absent, there will be excess alpha globin chains, and they will be precipitated in the erythroid cells, and that is the primary reason for the ineffective erythropoiesis and extramedullary hematopoiesis, all that you see in this patient. Before doing molecular diagnosis, it's very important to do some key laboratory diagnosis tests for thalassemias. And this first figure that shows that how the diagnosis of heterozygous beta thalassemia and other types of thalassemia is done. Let's focus more on beta thalassemia. And what you see here is that on the left side, where you have to use MCV, MCH, and hemoglobin F and A2 levels, and then you can do the diagnosis of beta thalassemia carrier, and you can differentiate beta thalassemia carrier from normal. On the right side, the box shows the hemoglobin F when it is increased from 50 to 98%, the patient may have homozygous beta thalassemia. That has to be correlated with the patient's clinical phenotype. So if you see a patient with a transfusion-dependent low hemoglobin organomegaly and F is elevated so much, and this may be concluded as the patient may be having homozygous beta thalassemia. There are a lot of other variations, rare variations. We can't talk about that because of lack of time. Then the bottom, the bottom figure shows how HPLC is done to quantitate hemoglobin A2, hemoglobin F levels, and we can differentiate heterozygous beta thalassemia, homozygous beta thalassemia, and the rare combinations of, it's not rare, but other combinations of beta globin mutations that producing abnormal hemoglobins in combinations with beta thalassemia. Why I put them there? Because those patients also may show severe transfusion-dependent disease, and phenotypically, clinically, you may not be able to differentiate those cases. And now I'm going to say that why do we need molecular diagnosis? The most important reason is, like Dr. Vikram Matthew said, when there is a patient who's transfused and the clinically looks like thalassemia major, and we cannot do a variant analysis to see hemoglobin A2 or F levels for diagnosis. Therefore, molecular diagnosis is absolutely required. And sometimes in the routine hemoglobin analysis by HPLC, we'll see unusual levels of hemoglobin F and A2 levels. And it's important to do a molecular diagnosis and find out what exactly is the mutation. We have to confirm and tell the clinician that the patient has homozygous beta thalassemia to carry on with the treatment. And the third reason is that for prenatal diagnosis, as you know, we cannot take fetal tissue and do an HPLC or hemoglobin analysis to see whether the patient has beta thalassemia or not. So you can extract DNA and do molecular diagnosis. And there are other reasons when you have a large number of cases, why some patients have severe disease, why some patients have mild disease. We can identify those mutations and correlate and see why some patients are transfusion dependent and why some patients become transfusion dependent later in life. And next slide, sir. So I'm going to take you through some of the methods that are used for molecular diagnosis. The left diagram shows the most commonly used method for detecting common mutations. Fortunately, there are some 7 mutations in the Indian population that are present in 80 to 90% of cases in the Indian population. Therefore, we can design the assays to detect these mutations rapidly. And this is a method called reverse dot plot, in which you can see the dots that are formed because of the PCR products that is amplified from the beta globin of the patient and hybridized. And there are some specific probes that can detect the mutations. And if you have any questions on this technique, we can answer those in the end. And the middle one is a technique called ARMS, Amplification Refractory Mutation System, where specific primers are used in the PCR, where these primers bind to the mutant sequence. And if it is amplified, then we'll tell that, okay, that mutation is present. And PCR products are run it on a gel and the diagnosis is done. The right side, the figure shows a common mutation that is seen in from patients from Sindhi population, where there's a large deletion of 600 base pair is deleted from the beta globin gene, and you can do a PCR and run it on a gel. When a small fragment is formed, we can tell that the patient has got 619 base pair deletions. And these methods can detect 80 to 90% of the cases of beta thalassemia, depending on the patients are from the south or north or west or east. This frequency can vary. Now, however, in a center like ours, where we do a comprehensive analysis, we do a mutation analysis for all the patients, we have to see patients whether they have rare mutations or not. For that, a method called DNA sequencing is used. In this protocol, the beta globin gene entire beta globin gene is amplified, then PCR is done, then DNA sequencing is carried out. The, the center of the figure, what you see is a DNA sequencer in which the products, the PCR products are mixed with a reagent that helps in the DNA sequencing are loaded. And the extreme right, the figure that shows that A, T, G, C, the 4 bases of the DNA can be separated and the mutations can be identified. And this is an example. The top panel shows there are 2 peaks there coming, that is from C and A, saying that the patient is heterozygous, and the bottom shows the patient from the normal individuals for comparison. So in short, the common mutations can be identified by a variety of methods. Subsequently, if the mutations are absent, we do DNA sequencing. Recently, we developed a new method for rapid detection of 13 common mutations, taking the patient's data from all over India. And this method was established in collaboration with Cgene. It's a company in Korea that makes such kits for detection of mutations. And this kit was developed exclusively for Indian patients. And here you can take a direct blood sample and add it to the tube. And the tube is kept in the real-time PCR machine. There are primers and probes to detect different mutations. And you can get the data which can be easily interpreted. And this test can be completed in 4 hours. A large number of samples can be analyzed every day if you have a large number of patients visiting your center, and it's highly sensitive, and we have analyzed around 500 cases so far, and we did not get any false positivity or false negativity. And it's much cheaper than any other method, and it costs only 450 rupees per sample. This is the spectrum of mutations that we have from analyzing more than 20,000 samples in our lab. And you can see here on the left different types of mutations that you can see in the exon 1, exon 2, exon 3 that we see very frequently. The right table shows the most common 13 mutations and their percentages. And these are the mutations that are included in the Cgene technology. Next slide, sir. So I also said that there are other type of mutations which we can't ignore, and they will be co-inherited with beta thalassemia. It's a different type of thalassemia where you can see here there's large deletions. The top panel is the beta globin cluster, where all the type of beta globin like genes are present in the beta globin cluster. And you can see the bars in the bottom of the gene, the cluster, that shows the extent of deletions that can occur in the beta globin cluster. When these deletions occur, more than one gene may be deleted: beta, delta, and or A gamma globin gene. So A gamma is expressed in the fetal stage. And these cases can be diagnosed if we do it very carefully. If the patient has got thalassemia major phenotype, and if you do the SPLC in the parents, you can easily identify by observing high hemoglobin F in one of the parents. And you can see in the left chromatogram showing high hemoglobin F, and that is not expected. It's around 25%. That's not usually expected in the normal individuals. And even heterozygous beta thalassemia, it can be much lower. It's around maximum we expect in heterozygous beta thalassemia is 2 or 3%. And the right side is a homozygous delta beta thalassemia. There is no hemoglobin A2. There is no hemoglobin A. A2 is absent because delta gene is also deleted. And the right side, the figure shows the HbF levels that can be used for differentiating delta beta thalassemia and hereditary persistence of fetal hemoglobin. And to detect these deletions, we have established a very easy method, very robust method, and it worked always in our lab. This is by using quantitative PCRs. Here, we amplify the gamma globin gene, delta and beta globin gene by fluorescently labeled primers, and we do this PCR and run it on a sequencer. What happens in the sequencer is not sequencing. What happens in the sequencer when we put this PCR product is this amplified PCR products are separated, and the peak heights can be estimated, and you can normalize these values and find out if there's a reduced peak height. Then we can say that delta, beta, and gamma are deleted or not. And this method is very, very robust. And you can see the graph on the right side, which detects that exactly zero 0.5 when the patient has got a heterozygous deletion of delta and beta. And we have done in the multiple samples in different types of mutations, we could quantitate it accurately. And this is the data. And as I said earlier, it's 100% sensitive and highly reproducible. When these deletions are absent, then we also do an MLPA. It's a probe-based method, and this kit can be obtained from MRC Holland, and it can be easily set up in the lab. And that this data shows that how MLPA data comes up and shows that the extent of deletions in the beta globin cluster can be detected. And the middle figure, that you can see that there is half height that shows that that much region of beta globin cluster is deleted, and the lowermost you can see the deletion is a bit larger and more parts of the beta globin cluster is deleted. So by using these methods, we can detect all sort of deletions that can occur in the beta globin gene. This is the spectrum of delta beta and HPF mutations that we have identified, and you can see here the extent of deletions. And in a center like ours, we do the molecular diagnosis for every case. It is very, very important to establish these methods. And as I said, 20,000 beta thalassemia cases, and we have done more than 2,000 PND cases accurately. We never had any issues at all of any misdiagnosis. And in 12 cases, only in 12 cases, we could not find the mutations out of this 20,000 beta thalassemia cases we studied so far. And this is my email ID, and my colleague who has done a lot of work along with me is Dr. Eunice. You can write to one of us if you need any details of what exactly is happening, or if you have any technical questions or the methods. And there's a paper which we published long time back in 2008, looking at different types of mutations in different regions of the country. And the lowermost, we can see here how we identified the novel deletions that can cause delta beta thalassemia and HPFH. I know that this technique sounded very complex. However, these techniques now, they are much, much easier to do. Establishing them was difficult, but now doing it in any other lab does not need a lot of time for the training. Thank you.
Thank you, Dr. Shaji. Congratulations for the lovely work you're doing. I think we need more centers like this to analyze the deletions and the mutations that happen in thalassemia all over India. We'll now go on to the management of thalassemia and to throw light on the issues related to multiple transfusions. Let me introduce to you Dr. Shivaraman. Shivaram C is going to talk about standard of care guidelines in thalassemia. He's the consultant and head of transfusion medicine, Manipal Hospital Transfusion Services.